Today we are going to see about gradient theory and factors affecting gradient. Gradient. A gradient is defined as a proportional rise and fall of certain factors in the embryo, the interaction of which bring about normal development. The word gradient is derived from the Latin word gradi, means to stop. The early development of embryo is controlled by one or more factors called gradients. These factors are distributed in a certain proportion in the embryo, with their rise at one end and fall at another end. This is called gradient. The gradient controls the normal development. Types of gradient theory. There are two types of gradient theory. They are single gradient theory, double gradient theory. Single gradient theory. This theory was proposed by C.M. Child in 1914. He shows that development and morphogenesis are controlled by a single factor is called single gradient or metabolic axial gradient. The oxidative metabolism is higher at the animal pole and it gradually decreases towards the vegetal pole along the median axis. The extensions of this gradient is proved by the reduction of Janus green. This theory says that the development of head structure is controlled by a high metabolic rate and the development of tail structure is due to the low metabolic rate. Double gradient theory. This theory was proposed by Rundstrom and Horstrandium in 1928. This theory states that the sea urchin egg has two factors or principles. They are animal gradient and the vegetal gradient. The animal gradient has its tender activity at the vegetal pole. The animal gradient diminishes towards the vegetal pole and the vegetal gradient diminishes towards the animal pole. They overlap throughout the whole egg. They are mutually antagonistic yet they interact. The normal development is due to a certain equilibrium between the two gradients. This system of gradient is called double gradient theory. According to this theory, the normal development depends upon the presence of both gradient and the equilibrium between them. Experimental evidence. The extensions of the double gradient theory and its mechanism can be proved in two ways: isolation and recombination of blastomeres, physicochemical method. Isolation and recombination of blastomeres. Rundstrom and Horstrandium carried out a series of experiments in sea urchin embryo to illustrate the extension of gradient system. They are as followed. Experiment number 1. The first two cleavages are meridional and they produced 4 equal blastomere. When the 4 blastomere are separated, each blastomere develop into normal larva. This is because of all the blastomere receive equal both animal pole material and the vegetal pole material. Experiment number 2. The third cleavage is equatorial. It produces 8 blastomere at this stage. The egg is cut into an animal half and the vegetal half. Both the half cannot develop into a normal larva, but they develop into a defective embryo. The animal pole developed into an animal pole structure only. This embryo is called animalized embryo. The animalized embryo consists of an ectodermal vesicle with a covering called cilia. The vegetal half develop into only a vegetal pole structure and the embryo is called vegetalized embryo. The vegetalized embryo has a large cut without a mouth and the falls developed into a normal embryo. This is due to the absence of animal gradient in the vegetal half and the vegetal gradient in the animal half. It is a picture for experiment number 2. Experiment number 3. In the 64 cell stage, the embryo has 3 types of blastomere, namely micromeres, macromeres, mesomere. They are arranged as 5 tiers. They are follows. The 2 tiers of mesomere situated at the animal pole, they are called AN1 and AN2. The 2 tiers of macromeres situated at the vegetal pole, they are called VEG1 and VEG2. 1 tiers of micromere at the vegetal pole. In an experiment, the different types are isolated and they are allowed to develop separately and in combination. The results are as follows. The animalized 1 alone developed into animalized embryo. The animalized 2 alone developed into animalized embryo. The vegetalized 1 is developed alone to develop vegetalized embryo. Again, the vegetalized 2 alone developed into a vegetalized embryo. The micromeres develops mesenchyma cell. The animalized 1 plus micromere are developed into animalized embryo. The animalized 2 plus micromere developed into animalized embryo. The vegetalized 1 plus micromere are developed into vegetalized embryo. The vegetalized 2 plus micromere developed into vegetalized embryo. Animalized 1 plus animalized 2 plus vegetalized 1 plus micromere are developed in normal larva. The vegetalized 1 plus vegetalized 2 plus animalized 1 plus animalized 2 are developed into a normal larva. It is picture for experiment number 3. Next, we are going to see about physicochemical experiment. The presence of gradient system in the developing embryo is determined by physicochemical experiment. Horst Strontium in 1952 carried out a fantastic experiment on CRG, plastula. CRG embryo are stained in Janus green, diethyl safran, dimethyl The stained embryo are placed in the dish sealed with a petroleum gel. The free oxygen in the dish is used by the embryo. After that, the embryo respires using the Janus green as the respirator of hydrogen. As a result, the blue Janus green is reduced as a red dye called dimethyl safran and then to a colorless substance called leucosaffrin. The reduction of Janus green followed a very characteristic sequence. The change in color blue into red is the first seen at the vegetal pole. Then the red color spreads out gradually and reaches the equator. At this experiment, at the extreme animal pole, the blue color changed into red and gradually spread towards the equator. When the blue color disappears completely, the red color begins to fade in the same sequence, starting first at the vegetal pole. This sequence of color change is definitely owing to the extensions of animal and vegetal gradient. Experiment number 2. The Hastomium separate the animal half and study the change in color. In the animal half, the animal gradient is predominant and the animalized embryo start reducing the Janus green at the animal pole first and then change the color separate towards the vegetal pole. There is no center of color change at the vegetal pole. Construct another experiment. Similarly, in the isolated vegetal half and the vegetal gradient is predominant and the vegetalized embryo start reducing the Janus green at the vegetal pole first and then the change in colors separate towards the animal pole. Here there is no center of color change at the animal pole. The above experiment clearly indicate that the extension of gradient are due to the extent of metabolic reaction which involve oxidation. The metabolic rates are higher at the poles and decrease towards the equator or in the opposite direction. Next, we are going to see about factors affecting gradients. There are large number of factors, especially chemical agents, which affect the gradient system, thereby modifying the normal morphogenesis. The chemical may suppress the animal gradient or the vegetal gradient. Animalizing agent. The certain chemical cause predominant development of animal pole structure. These chemicals are called animalizing agent and the embryo is called animalized embryo. Mode of action of animalizing agent. The animalizing agent either attack the protein or form common with the protein. They block the active site of protein, thus inhibiting them from interacting with other cell components. Vegetalizing agents. Certain chemicals cause predominant development of vegetal pole structure. These chemicals are called vegetalizing agent and the embryo is called vegetalized embryo. The vegetalizing agents are as follow. Seawater containing some lithium salt, dinitrophenol, sodium acid, etc. Mode of action of vegetalizing agent. The vegetalizing agent like sodium acid and dinitrophenol are enzyme poison. Acid inactivates the cytochrome oxidase system while the dinitrophenol disturb respiration by preventing oxidative phosphorylation. When there is no oxidative phosphorylation, the ATP molecule cannot be produced. Hence, the vegetation is brought about in the embryo by the disturbance of oxidative process or by the disturbance of process of phosphorylation. Thank you.